The goal of this video is to talk about the TCA cycle, also known as the Krebs cycle, also known as the citric acid cycle. Most importantly, this cycle is essentially the hub of all of the metabolic processes that occur in the body. So in class, we've already talked about how acetyl-CoA is generated by the pyruvate dehydrogenase complex, and we will review that material a little, but the major focus of this video is going to be the reactions of the TCA cycle. We're going to talk about the mechanisms of those reactions, how they occur, and these mechanisms are going to be really important to understand because one of our goals for understanding the TCA cycle is to be able to track specific carbons as they move through the cycle. In addition, we're also going to talk about the control mechanisms that regulate the TCA cycle to determine how materials go through the TCA cycle, and then finally we'll summarize what you've learned. One thing that's really important is because this is an online video, you should go through it at your own pace. Make sure to pause the video to take notes. Also, feel free to rewind as you need to understand the material. So now let's start by defining the TCA cycle. So the TCA cycle is a cycle of 8 enzymes, you can see here. And this basically generates the precursors for lots of biosynthetic pathways. And in addition, lots of biosynthetic pathways can also feed into the TCA cycle. For example, breakdown of many amino acids feeds into the TCA cycle, just as we learned about oxaloacetate when we were talking about gluconeogenesis. So this is a really important cycle to cellular metabolism, and it integrates both biosynthetic pathways as well as pathways that are used to burn glucose for energy. So acetyl groups enter the TCA cycle as acetyl-CoA, as we learned in the last class. So acetyl-CoA, as you remember, is produced by the pyruvate dehydrogenase complex. As you recall, this is an example of an oxidative decarboxylation reaction because a hydride is transferred to produce a molecule of NADH, and also one carbon is released as CO2. So this goes from a 3-carbon pyruvate molecule to a 2-carbon acetyl-CoA molecule. And the acetyl group is shown here, and this is a way to transfer energy. So the acetyl group contains this high energy thioester bond, which has an energy. Hydrolysis of this bond releases about 31.5 kilojoules per mole. And we're actually going to see this CoA compound again, but this is a way to transfer energy. So just to quickly review the pyruvate dehydrogenase complex, as you remember, this is a really large multi-enzyme complex, and it's composed of 3 different enzymes that are non-covalently associated with one another. So E1 is the pyruvate dehydrogenase enzyme, E2 is dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. So this is a very large complex, but together it accomplishes this reaction where it converts pyruvate to acetyl-CoA, producing a molecule of NADH and releasing a molecule of carbon dioxide. So just to recall, these 3 enzymes actually accomplish 5 different reactions in sequence, which requires 5 different either coenzymes or prosthetic groups. So first, in E1, TPP, which we've already learned, can stabilize these very unstable ketone carbanions. So in the first reaction, TPP is used to stabilize this ketone carbanion, which catalyzes the release of CO2, and we form this hydroxyethyl TPP conjugate. So this hydroxyethyl TPP, so this is essentially our acetyl group, or it's going to be our acetyl group. Then from TPP, this group is transferred to the lipoamide arm of E2. So now we have this acetyl group that is bound to this dihydrolipoamide arm, and as you remember, this is a really long chain that can then reach into E3. So the lipoamide arm transfers the acetyl group to CoA. So now we've made our product acetyl CoA, but we have a problem because the lipoamide arm can be reversibly oxidized and reduced. And here it's reduced, so we have to reoxidize this lipoamide arm. That's accomplished through the action of FAD NE3. So FAD then reoxidizes this lipoamide arm so that it can act again to take that acetyl group from hydroxyethyl TPP. But now we actually have reduced FAD. So in order to reoxidize FADH2 to FAD, in this case we have NAD. So NAD is reduced in order to reoxidize FAD. So now we have NADH, which is our final product. We also have acetyl CoA, and we've produced a molecule of CO2. So now we've produced all three of our products using several different coenzymes and prosthetic groups. So TPP to stabilize that ketone carbanion, the dihydrolipoamide arm, which serves two functions. So one, it serves as an arm that can reach all the way from E1 to E3, and it also oxidizes the carbanion to form the acetyl group, and it transfers that acetyl group to CoA, which is another one of our coenzymes. The function of FAD was to reoxidize the lipoamide arm, and the function of NAD was to reoxidize FADH2 to FAD. So just as to briefly review, this the pyruvate dehydrogenase complex is regulated primarily by product inhibition. So these reactions can actually go in either direction because they operate near equilibrium. But if we accumulate too much acetyl CoA or too much NADH, this actually drives the reaction in the backwards direction to form pyruvate. This makes sense because, as you recall, pyruvate can then undergo gluconeogenesis to form glucose if we have very high energy conditions. In addition, the activity of E1 can also be regulated by phosphorylation, and importantly, the enzymes that regulate this phosphorylation are also regulated by sensors of energy within the cell. Okay, so now we're ready to move into the actual TCA cycle. So we're going to begin with reaction 1. So in reaction 1, the first enzyme is citrate synthase. So citrate synthase converts oxaloacetate, which is a 4-carbon compound. It adds the 2 carbons from the acetyl group from acetyl CoA to make the 6-carbon citrate molecule. So we're combining 4-carbon citrate plus 2 carbons from acetyl CoA to form 6-carbon citrate, and we release a molecule of CoA. So to take a look at this mechanism, much like what we saw for hexokinase, this is an enzyme that has a cleft, that the active site lies within a cleft, and that the conformation of the enzyme closes. It's approximately an 18-degree shift to close that enzyme to make sure that we don't have any side reactions that can occur. So in the open conformation, the enzyme can bind oxaloacetate, but it can't bind acetyl CoA. And remember, acetyl CoA was what has that very high energy CoA bond. So this makes sure that we don't have non-productive hydrolysis of CoA. So once oxaloacetate is bound, the enzyme closes, and this generates the binding site for acetyl CoA, which allows the reaction to go forward. Okay, so the first thing that happens in the citrate synthase mechanism is that aspartic acid 375 is going to act as a general base, and it generates the enolate ion of acetyl CoA. It's important to note that this is actually the rate limiting step in this case. So here you can see now we've generated this enolate ion. So we have the double bond between the two carbons and the negatively charged oxygen. It's likely that this histidine 274 forms a hydrogen bond here in order to help stabilize this enolate ion. But here, the double bond is going to act as a nucleophile to attack the carbonyl carbon of oxaloacetate. This pushes the electrons out onto the oxygen, and His 320 can then donate a proton to this to form the hydroxyl group here. So this generates citral-CoA, as you can see here. So now we have our CoA is covalently bound to the carbon. This carbon is now a tetrahedral carbon, which is also which now has an alcohol attached to it instead of the carbonyl. So then in the next step, this citral-CoA is going to be hydrolyzed, and this releases CoA. We're not going to talk in detail about this mechanism, but just note that because it's very energetically favorable to hydrolyze this CoA group, CoA is easily released. So now we have citrate, and what's important to note about citrate is that this is not a chiral molecule. So I want you to turn off the video and think for a minute how we know that this is not chiral. Okay, so how you know this isn't chiral is because both of these, or if we look at this carbon, both of these arms are identical. So we don't have 4 different substituents attached to this tetrahedral carbon, we only have 3 different substituents. But because we have 3, this is what's known as a prochiral molecule. Because if we made one change here, this would allow this molecule to become chiral. What's really important here to recognize is, if you remember, lots of times enzymes can hold molecules in such a way that a prochiral molecule can generate only one chiral product, and that's going to become important later. Okay, so now let's talk about reaction 2. So reaction 2 is the aconitase reaction. So aconitase is going to convert citrate to isocitrate. And it's going to go through this cis-aconitate intermediate. So even though citrate is not a chiral molecule, it is prochiral. And because this is prochiral, and aconitase holds it in such a way to act as if it's chiral, this reaction is stereospecific and will only produce one product. Okay, so in the aconitase mechanism, the first thing you'll note is that we have this iron-sulfur cluster. We're going to see several of these, particularly in the electron transport chain, and they're going to serve different functions, but they're all involved in some type of metal ion catalysis. And here the role of the iron is to stabilize the negative charge on the carboxyl groups. So in the mechanism, the first thing that happens is that serine 642 here is going to act as a general base. You'll note this is a little unusual, but this works in a way that's very similar to what we've already learned for the serine protease mechanism. So in this mechanism, serine 642, this oxygen has lost its hydrogen, and it's going to act to extract a proton from citrate. That causes the electrons to come in, form a double bond between the two carbons, which then kicks off this hydroxyl group. The hydroxyl group can then associate with this proton from histidine 101. This histidine and this aspartic acid form a hydrogen bonded network. Once again, similar somewhat to a catalytic triad, except here we only have a dyad. So now in the next step, you can see we formed this cis-aconitate intermediate. So we have the double bond between the two carbons here, and then we have two carboxyl groups as well as a carboxyl group here. So something strange happens in the mechanism here. The enzyme actually changes conformation, which drives a 180-degree flip for this cis-aconitate intermediate. So C2, which was located down here and wasn't associated with the iron sulfur cluster, now flips into this position, and now you can see that C2, that carboxyl group, is now being stabilized by the iron sulfur cluster, and now we have our C3 sitting at the other end of the enzyme. So this 180-degree flip now allows our water molecule, which remember is being stabilized by this histidine aspartic acid dyad, now the alcohol from that water molecule, this hydroxyl group, can act as a nucleophile to attack this carbon. So whereas it was once attached to C3, now it's going to attack C2, which forces the electrons from the double bond out, and these electrons will then extract the proton from serine 642 for pushing these electrons back to the oxygen. So in this case, serine 642 has now acted as a general acid. So we've regenerated serine 642 in the active site, and now our hydroxyl group is attached to C2 instead of C3. And that's the end of this video. The next reaction, reaction 3, will start with the next video.